Hello everyone, welcome to the next lecture of our physiology video lecture series. This lecture is titled Gastrointestinal Physiology. In this particular system, we will delve in detail into the gastrointestinal system and learn about how it functions and how it is regulated. So let us get started. First, we'll look into the general principles of the gastrointestinal function. Here we have a schematic which shows us the various organs and organ systems that completely make up the gastrointestinal system. The alimentary tract provides the body with a continual supply of water, electrolytes, and nutrients via intake through the mouth. To achieve this requires movement of food through the alimentary canal or the alimentary tract, which runs from through our mouth across lengthwise across our body right to the anus. The movement thus to provide continual supply of water, electrolytes, and nutrients, there needs to be a movement of food through the alimentary tract, secretion of digestive juices and digestion of food, absorption of water, various electrolytes and digestive products, circulation of blood through the gastrointestinal organs to carry away the absorbed substances, control of all these functions by local nervous and hormonal systems. The figure here shows the entire alimentary canal. Each part is adapted to its specific functions. Some to simple passage of food, such as the esophagus, which just allows the transfer of food through it from the mouth towards the stomach. Others to temporary storage of food, such as the stomach, and others to digestion and absorption, such as the small intestine. Let us first look into the physiologic anatomy of the gastrointestinal wall. So the figure here shows the typical cross section of the intestinal wall, which is shown right here in this section. So what this means is that if the alimentary canal was taken lengthwise and was just cut across its length, then this is what we will see. Layers of muscles that are present which are different from one another. These different layers include, from the outer surface inward, the serosa, a longitudinal muscle layer, a circular muscle layer, a submucosa, and the mucosa. In addition, sparse bundles of smooth muscle fibers, which are called the mucosal fibers or mucosal muscles, lie in the deeper layers of the mucosa. The motor functions of the gut are performed by different layers of the smooth muscle. It is to be noted that the gastrointestinal smooth muscle functions as a syncytia. That is, the whole gastrointestinal smooth muscle system contracts or relaxes at once, as if it is acting as a single muscle. Smooth muscle fibers in the gastrointestinal tract are arranged in bundles of as many as 1,000 parallel fibers. Within each bundle, the muscle fibers are electrically connected with one another through large number of gap junctions that allow low resistant movement of ions from one muscle cell to the next. Therefore, each muscle layer functions as a syncytia. That is, when an action potential is elicited anywhere within the muscle mass, it generally travels in all directions in the muscle. Also, a few connections exist between the longitudinal and the circular muscle layers, so that excitation of one of these layers often excites the others as well. Thus, excitation along anywhere in the muscle mass will cause the whole muscle mass of the gastrointestinal wall to contract and relax together, and thus we can say that the gastrointestinal wall acts as a syncytium. Next, we look into the electrical activity of the gastrointestinal smooth muscle. The smooth muscle of the gastrointestinal tract is excited by almost continual, slow, intrinsic electric activity along the membranes of the muscle fibers. This activity has two basic types of electrical waves, the slow waves and the spikes. Most gastrointestinal contractions occur rhythmically, and this rhythm is determined mainly by the frequency of the so-called slow waves of the muscle membrane potential, which are shown in the schematic right here. In this particular plot, we have plotted the membrane potential in millivolts against seconds, and we can see the first part shows us the resting rhythmical slow waves that are present in the gastrointestinal smooth muscles. These waves shown in the figure are not action potentials but slow undulating changes in the resting membrane potential. They appear to be caused by interactions among the smooth muscle cells and specialized cells called the interstitial cells of Cajal, which act as electrical pacemakers for the smooth muscle cells. The slow waves usually do not by themselves cause muscle contraction in most part of the gastrointestinal tract, except perhaps the stomach. Instead, they mainly excite the appearance of intermittent spike potentials, and the spike potentials in turn actually excite the muscle contraction. Thus, the slow waves are essentially facilitating the smooth muscle, gastrointestinal smooth muscle wall. Facilitation is something that we have just looked into in the last lecture, and I would recommend you to just go look at it if we are having for problems recalling what facilitation really is. Next, we look at the spike potentials. The spike potentials are the true action potentials. They occur automatically when the resting membrane potential of the gastrointestinal smooth muscle becomes more positive than about minus 40 millivolts. The normal resting membrane potential in the smooth muscle fibers of the gut is between minus 50 to minus 60 millivolts. And thus, during a spike potential, the membrane potential rises to above minus 40 and action potential is generated. Thus, note in the figure that each time the peaks of the slow waves temporarily become more positive than minus 40 millivolts, that is right here, spike potentials appear on these peaks. In gastrointestinal smooth muscle fibers, the channels responsible for the action potentials are called the calcium-sodium channels. These channels are much slower to open and close than the rapid sodium channels of large nerve fibers. The slowness of opening and closing of calcium-sodium channels account for the long duration of the action potentials. So these are the action potentials or the spikes that are shown. The spikes are divided into three parts in this particular plot, in which first is stimulation by stretch, that is the stimulation of the gastrointestinal wall when the gastrointestinal wall muscles are stretched. The second is stimulation by acetylcholine, which is more or less similar. And finally, we have a stimulation by parasympathetics, that is the parasympathetic nerve system, which helps along the digestion, or it helps along the digestion path. That we know that the parasympathetic nervous system, when activated or stimulates the body in order to activate the rest and digest action, and thus the parasympathetics essentially help in digestion, and we see a huge rise in spikes when the gastrointestinal muscle wall is stimulated using parasympathetics. Next, we have stimulation by norepinephrine and sympathetics. Norepinephrine and sympathetics are obviously preparing the body for the flight or fight response, and thus resting responses such as digestion are completely stopped, and hence we see a hyperpolarized state of the gastrointestinal muscle wall, that is digestion and movement along the gastrointestinal wall and its contraction are completely held at a stop. Next, we look into the neural control of the gastrointestinal function, which is performed by the enteric nervous system. The gastrointestinal tract has a nervous system all on its own called the enteric nervous system. It lies entirely in the wall of the gut, beginning in the esophagus and extending all the way to the anus. The enteric nervous system is composed mainly of two plexuses, that is, two layers. An outer plexus lying between the longitudinal and circular muscle layers called the myenteric plexus, and an inner plexus called the submucosal plexus. Both of them are shown right here, with the myenteric plexus being further away from the epithelium layer and the submucosal plexus being closer. The myenteric plexus controls mainly the gastrointestinal movements, and the submucosal plexus controls mainly gastrointestinal secretion and local blood flow. The extrinsic sympathetic and parasympathetic fibers that connect to both the myenteric, the extrinsic sympathetic and parasympathetic fibers connect to both the myenteric and submucosal plexuses, and thus the gastrointestinal function is regulated by the central nervous system via the enteric nervous system. It is to be noted that even during development, the enteric nervous system develops entirely differently from the normal central nervous system that develops in all organisms. Thus, the enteric nervous system is essentially also develops separately. Although the enteric nervous system can function on its own, independently of these extrinsic nerves, stimulation by parasympathetic and sympathetic systems can greatly enhance or inhibit gastrointestinal functions. When the myenteric plexus is stimulated, its principal effects are increased tonic contraction or tone of the gut wall, increased intensity of the rhythmical contractions, slightly increased rate of rhythm of contraction, increased velocity of conduction of excitatory waves along the gut wall, which causes more rapid movement of the gut peristaltic waves. Peristaltic waves are the waves of the gut muscles which push the bolus or the food along the alimentary canal, and something that we'll look into in later slides. The submucosal plexus is mainly concerned with controlling function within the inner wall of each minute segment of the intestine. For example, many sensory signals are integrated in the submucosal plexus to help control local intestinal secretion, local absorption, and local contraction of submucosal muscle. Next, we look into in detail the control of the gastrointestinal tract by nervous and hormonal control. First, we look into the autonomic control of the gastrointestinal tract. Obviously, we know that the gastrointestinal system is not under our voluntary control, and function autonomously. And thus, the autonomous nervous system is the one that controls it. So we'll look into the autonomic control of the gastrointestinal tract. These are obviously divided into the two major halves, the parasympathetic control and the sympathetic control. In the parasympathetic innervation, stimulation of the parasympathetic nerves causes general increase in activity of the entire enteric nervous system. This in turn enhances activity of most gastrointestinal functions. Next, we have the sympathetic innervation. In general, stimulation of sympathetic nervous system inhibits activity of the gastrointestinal tract, causing many effects opposite to those of the parasympathetic system. Strong stimulation of the sympathetic system can inhibit motor movements of the gut so greatly that this literally can block movement of the food through the gastrointestinal tract. After the autonomic control, we will look into the hormonal control of gastrointestinal motility. One of the hormonal control obviously includes the hormones which control the gastrointestinal motility, and we will discuss various hormones that are secreted by the gut wall that control the same. The first one we will look into is gastrin. Gastrin is secreted by the G cells of the antrum of the stomach in response to stimuli associated with ingestion of a meal, such as distension of the stomach, the products of proteins, and gastrin-releasing peptide, which is released by the nerves of the gastric mucosa during vagal stimulation. The primary actions of gastrin are stimulation of gastric acid secretion and stimulation of growth of the gastric mucosa. Next, we will look into the other hormones, which include cholecystokinin, which is secreted by the eye cells in the mucosa of the duodenum, and the jejunum, mainly in response to digestive products of fat, fatty acids, and monoglycerides in the intestinal contents. This hormone strongly contracts the gallbladder, expelling bile into the small intestine, where the bile in turn plays important roles in emulsifying fatty substances, allowing them to be digested and absorbed. Next, we look into secretin, which is secreted by the S cells in the mucosa of the duodenum in response to acidic gastric juice emptying into the duodenum from the pylorus of the stomach. Secretin has a mild effect on motility of the gastrointestinal tract and acts to promote pancreatic secretion of bicarbonate, which in turn helps to neutralize the acid in the small intestine. Next is the gastric inhibitory peptide, which is secreted by the mucosa of the upper small intestine in response to fatty acids, amino acids, and carbohydrate. It has a mild effect in decreasing motor activity of the stomach and therefore slows emptying of the gastric contents into the duodenum when the upper small intestine is already overloaded with food products. The final hormone we'll look into is motilin, which is secreted by the upper duodenum during fasting. And the only known function of this hormone is to increase the gastrointestinal motility. Next, we look into the functional types of movements in the gastrointestinal tract. The two types of movements which occur in the gastrointestinal tract are the propulsive movements, which cause food to move forward along the tract at an appropriate rate to accommodate digestion and absorption, and mixing movements, which keep the intestinal contents thoroughly mixed at all times. Here we can see the propulsive movements, that is the peristalsis, where at zero time there is, when the peristaltic contraction occurs, there is a leading wave of distension that after some time extends along the gastrointestinal tract. The basic propulsive movement of the gastrointestinal tract is peristalsis, which is illustrated in the figure. A contractile ring appears around the gut and then moves forward. This is analogous to putting one's fingers around a thin distended tube, then constricting the fingers and sliding them forward along the tube. Any material in front of the contractile ring is moved forward. The usual stimulus for intestinal peristalsis is distension of the gut. Other stimuli that can initiate peristalsis include chemical or physical irritation of the epithelial lining of the gut. Also, strong parasympathetic nervous signals to the gut will elicit strong peristalsis. Next, we look into the function of the enteric plexus in peristalsis. Peristalsis occurs only weakly or not at all if any portion of the gastrointestinal tract has a congenital absence of the enteric plexus. Congenital means from birth. Therefore, effectual peristalsis requires an active enteric plexus. Next is that the direction of movement of the peristaltic waves are towards the ends. Peristalsis theoretically can occur in either direction from a stimulated point, but it normally dies out rapidly in the oral direction, that is towards the mouth, while continuing for a considerable distance towards the ends. At the same time, the gut sometimes relaxes several centimeters downwards towards the anus, which is called receptive relaxation, thus allowing food to be propelled more easily anally than orally. Next, we look into the second type of movement that we are talking about, which is the mixing movements. Mixing movements differ in different parts of the alimentary tract. In some areas, the peristaltic contractions themselves cause most of the mixing. This is especially true when forward progression of the intestinal contents is blocked by a sphincter, so that a peristaltic wave can then only churn the intestinal contents rather than propelling them forward. At other times, local intermittent constrictive contractions occur every few centimeters in the gut wall. These constrictions usually last only 5 to 30 seconds. Then new constrictions occur at other points in the gut, thus chopping and shearing the contents first here and there. And thus food the entire food is chopped up into small pieces and mixed together for digestion to effectively occur. So let us continue with propulsion and mixing of food in the alimentary canal. This propulsion and mixing of food in the alimentary tract begins with the process of ingestion of food. The first step of ingestion of food is obviously chewing or mastication. The teeth are designed for chewing. The anterior teeth, which are the incisors, providing a strong cutting action, and the posterior teeth, the molars, a grinding action. Much of the chewing process is caused by a chewing reflex, which can be explained as follows. The presence of a bolus of food in the mouth at first initiates a reflex inhibition of the muscles of mastication, which allows the lower jaw to drop. The drop in turn initiates a strength reflex of or the stretch reflex of the jaw muscles that leads to rebound contraction. This automatically raises the jaw to cause closure of the teeth, but it also compresses the bolus against the linings of the mouth, which inhibit the jaw muscles once again, allowing the jaws to drop and rebound another time. This process is repeated again and again. Chewing is important for digestion of all foods, but especially important for most fruits and raw vegetables because they have indigestible cellulose membranes around their nutrient portions that must first be broken down before the food can be digested. Chewing aids the digestion of food by breaking it down into smaller pieces to increase the total surface area exposed to the digestive secretions. The next step in the process is the swallowing or deglutition of the bolus of food. In general, swallowing can be divided into two separate stages. The first one is a voluntary stage, which initiates the swallowing process. The second is a pharyngeal state, which is involuntary and constitutes passage of food through the pharynx into the esophagus. And finally, we have the esophageal phase, which is another involuntary phase that transports food from the pharynx to the stomach. So let us look into the various stages. So let us look into the various stages of swallowing in detail. The first stage is obviously the voluntary stage of swallowing. When the food is ready for swallowing, it is voluntarily squeezed or rolled posteriorly into the pharynx by the pressure of the tongue upward and backward against the palate, as shown in this particular picture. From here on, swallowing becomes entirely, or rather almost entirely, automatic and ordinarily cannot be stopped. Next is the pharyngeal state of swallowing. As the bolus of food enters the posterior mouth and pharynx, it stimulates epithelial swallowing receptor areas all around the opening of the pharynx, especially on the tonsillar pillars. And impulses from these pass to the brainstem in order to initiate a series of automatic pharyngeal muscle contractions as follows. First, the soft palate is pulled upward to close the posterior nares to prevent the reflux of food into nasal cavities. Next, the palatopharyngeal folds on each side of the pharynx are pulled medially to approximate each other. In these ways, the folds form a sagittal slit through which the food must pass into the posterior pharynx. Next, the vocal cords of the larynx are strongly approximated, and the larynx is pulled upward and anteriorly by the neck muscles. These actions, combined with the presence of ligaments that prevent upward movement of the epiglottis, cause the epiglottis to swing backward over the opening of the larynx. All these effects acting together prevent the passage of food into the nose and trachea. The upward movement of the larynx also pulls up and enlarges the opening to the esophagus. At the same time, the upper 3 to 4 centimeters of the esophageal muscle wall, called the upper esophageal sphincter, which is also called the pharyngoesophageal sphincter, relaxes, thus allowing food to move easily and freely from the posterior pharynx into the upper esophagus. And finally, once the larynx is raised and the pharyngoesophageal sphincter becomes relaxed, the entire muscular wall of the pharynx contracts, beginning in the superior part of the pharynx, then spreading downward over the middle and the inferior pharyngeal areas, which propels the food by peristalsis into the esophagus. The next stage in the swallowing process is the esophageal stage of swallow. The esophagus functions primarily to conduct food rapidly from the pharynx to the stomach. The esophagus normally exhibits two types of peristaltic movements: primary peristalsis and secondary peristalsis. Primary peristalsis is simply the continuation of the peristaltic wave that begins in the pharynx and spreads into the esophagus during the pharyngeal stage of swallowing. If the primary peristaltic wave fails to move into the stomach all the food that has entered the esophagus, secondary peristaltic wave will result from distension of the esophagus itself by the retained food. These waves continue until all the food has finally emptied into the stomach. After the swallowing phase has been completed, we will now look in detail about the stomach and the functions of the stomach. The first thing we'll look into is the motor functions of the stomach. The motor functions of the stomach can be threefold: storage of large quantities of food until the food can be processed in the stomach, duodenum, and lower intestinal tract, mixing of these food with gastric secretions until it forms a semi-fluid mixture called the chyme, and slow emptying of the chyme from the stomach into the small intestine at a rate suitable for proper digestion and absorption by the small intestine. Next, we look into the function of the stomach in storing of the chyme or the. Normally, when food stretches the stomach, a vagovagal reflex from the stomach to the brain stem and then back to the stomach reduces the tone of the muscular wall of the body of the stomach so that the wall bulges progressively outward, accommodating greater and greater quantities of food up to a limit in the completely relaxed stomach of 0.8 to 1.5 liters. Next, we look into the mixing, propulsion, and emptying of the stomach. Mixing and propulsion of food in the stomach is based on the basic electrical rhythm of the stomach that we had seen in the previous slides. That is the slow waves that occur in the gastrointestinal system. As long as food is in the stomach, weak peristaltic constricted waves called mixing waves begin in the mid-upper portion of the stomach wall and move towards the antrum about once every 15 to 20 seconds. The antrum is obviously the angular bend that is present in the lower stomach. That is shown right here in this picture. This is the angular notch and this portion is known as the antrum. These waves, that is the mixing waves, are initiated by the gut wall basic electrical rhythm, consisting, as I said, of the electrical slow waves that occur spontaneously in the stomach wall. As the constrictor waves progress from the body of the stomach into the antrum, they become more intense, some becoming extremely intense and providing powerful peristaltic action potential, driven constrictor rings that force the antral contents under higher and higher pressure towards the pylorus. Thus, the moving peristaltic constrictive ring combined with the upstream squeezing action called retropulsion is an exceedingly important mixing mechanism in the stomach. Next, we look into the intense antral peristaltic contractions during stomach emptying, which is known as the pyloric pump. Most of the time, the rhythmical contractions of the stomach are weak and function mainly to cause mixing of food and gastric secretions. However, 20% of the time, while food is in the stomach, the contractions become intense, beginning in the mid-stomach and spreading through the caudal stomach, no longer as weak mixing contractions, but as strong peristaltic, very tight ring like constrictions that can cause stomach emptying. Next, we look into the regulation of stomach emptying. These are the first factor that is involved in the regulation of stomach emptying that we'll look into are the gastric factors. The first gastric factor is the effect of gastric food volume on rate of emptying. Increased food volume in the stomach promotes increased emptying from the stomach. Stretching of the stomach wall elicits local maintaining reflexes in the wall that greatly accentuate activity of the pyloric pump and at the same time inhibit the pylorus. Next is the effect of hormone gastrin on stomach emptying. Gastrin has mild to moderate stimulatory effects on motor functions in the body of the stomach. Most important, it seems, to enhance the activity of the pyloric pump. Thus, it too probably promotes stomach emptying. Next, we'll look into powerful duodenal factors that inhibit stomach emptying. The first factor we'll look into is inhibitory effect of enterogastric nervous reflexes from the duodenum. The types of factors that are continually monitored in the duodenum and that can initiate enterogastric inhibitory reflexes include the following. The first is the degree of distension of the duodenum. The second is the presence of any degree of irritation of the duodenal mucosa. Third is the degree of acidity of the duodenal chyme. Fourth is the degree of osmolality of the chyme. And finally, we have the fifth one, which is the presence of certain breakdown products in the chyme, especially of the proteins. Now we'll go into various parts of the gastrointestinal system and see how exactly movement along that particular part occurs. The first one that we'll tackle is the movements of the small intestine. First type of movement that we'll discuss is mixing contractions, which are called segmentation contractions as well. When a portion of the small intestine becomes distended with chyme, stretching of the intestinal wall elicits localized concentric contractions spaced at intervals along the intestine and lasting a fraction of a minute. The contractions cause segmentation of the small intestine. The maximum frequency of the segmentation contractions in the small intestine is determined by the frequency of electrical slow waves in the intestinal wall, which is the basic electrical rhythm. Next, we have the propulsive movements that occur in the small intestine, the first one of which is the peristaltic. Chyme is propelled through the small intestine by peristaltic waves. These can occur in any part of the small intestine and they move towards the anus at a velocity of 0.5 to 2 meters per second. Faster in the proximal intestine and slower in the terminal intestine. They normally are very weak and usually die out after traveling only 3 to 5 centimeters, very rarely farther than 10 centimeters. This means that 3 to 5 hours are required for passage of chyme from the pylorus to the ileocecal valve. Next is we look into the control of peristalsis in the small intestine by nervous and hormonal signals. Peristaltic activity of the small intestine is greatly increased after a meal, partly by stretch of the duodenal wall caused by the chyme entering it, but also by a gastroenteric reflex that is initiated by distension of the stomach and conducted principally through the Enteric plexus from the stomach down along the wall of the small intestine. In addition to nervous signals that may affect small intestinal peristalsis, several hormonal factors also affect peristalsis. They include gastrin, CCK, cholecystokinin, insulin, motilin, and serotonin, all of which enhance intestinal motility and are secreted during various phases of food processing. After being done with the movements of the colon, we now look, after being done with the movements of the small intestine, we look into the movements of the colon. The principal functions of the colon are: first is the absorption of water and electrolytes from the chyme to form solid feces, and secondly, storage of fecal matter until it can be expelled. The proximal half of the colon, as shown in the figure, is concerned primarily with absorption and the distal half with storage. First, we look into the mixing movements that occur in the colon, which are known as horstrations. In the same manner that segmentation movements occur in the small intestine, large circular constrictions occur in the large intestine. At each of these constrictions, about 2.5 centimeters of the circular muscle contracts. At the same time, the longitudinal muscle of the colon contracts. These combined contractions of the circular and the longitudinal strip of muscle causes unstimulated portion of the large intestine to bulge outward into bag-like sacs called horstrations. Next, we look into the propulsion movements or mass movements that occur. Much of the propulsion in the cecum and the ascending colon results from slow but persistent contractions, requiring as many as 8 to 15 hours to move the chyme from the eosinophil valve through the colon, while the chyme itself becomes fecal in quantity, a semi-solid slush instead of semi-fluid. From the cecum to the sigmoid, mass movements can, for many minutes at a time, take over the propulsive flow. A mass movement is a modified type of peristalsis characterized by following sequence of events. First, a constrictive ring occurs in response to a distended or irritated point in the colon, usually present in a transverse colon. Then rapidly the 20 or more centimeters of the colon distal to the constrictive ring lose their hastrations and instead contract as a unit, propelling the fecal matter in the segment en masse further down the colon. The contraction develops progressively more force for about 30 seconds and relaxation occurs during the next 2 3 minutes. Then another mass movement occurs, this time perhaps further along the colon. This is how finally food is pushed and propulsion and mixing of food occurs along the alimentary canal, starting from the mouth and finally ending in the large intestine. Next, we look into digestion and absorption in the gastrointestinal tract. So now we have, after having covered the propulsion and mixing of food along the alimentary tract, we'll now look into exactly how the digestion and absorption part of the process occurs. Digestion is essentially the hydrolysis of various nutrients present in food to smaller and smaller parts till they can be absorbed easily by the gastrointestinal lining. These includes the hydrolysis of carbohydrates, the hydrolysis of fats, and the hydrolysis of proteins mainly. First, let us look into the hydrolysis of carbohydrates. Almost all carbohydrates of the diet are either large polysaccharides or disaccharides, which are combinations of monosaccharides bound to each other by condensation. When carbohydrates are digested, the above process is reversed and carbohydrates are converted to monosaccharides. Specific enzymes in the digestive juices of the gastrointestinal tract return the hydrogen and the hydroxyl ions from water to the polysaccharide and thereby separate the monosaccharides from each other. This process of addition of water molecules to another macromolecule is known as hydrolysis. Next is the hydrolysis of fats. Almost the entire fat portion of the diet consists of triglycerides, which are a combination of 3 fatty acid molecules condensed with a single glycerol molecule. During condensation, 3 molecules of water are removed. Digestion of triglycerides is also a reverse of this process. The fat digestive enzymes return 3 molecules of water to the triglyceride molecule and thereby split the fatty acid molecule away from the glycerol. Here again, the digestive process is one of hydrolysis. Next is the hydrolysis of proteins. Proteins are formed from multiple amino acids that are bound together by peptide linkages. At each linkage, a hydroxyl ion has been removed from the one amino acid and a hydrogen ion has been removed from a successive one. Thus, successive amino acids in the protein chain are bound together by condensation, and digestion occurs again by the reverse effect, that is hydrolysis. The proteolytic enzyme return hydrogen and hydroxyl ions of the water molecule to the protein molecules to split them into their constituent amino acids. After looking briefly into how the digestion of the carbohydrates, fats, and proteins occur, we will now look in detail into the whole process. First, we look into the digestion of carbohydrates. The carbohydrates food of our diet consists of three major sources, which include sucrose, which is a disaccharide popularly known as cane sugar, lactose, which is a disaccharide present in milk, and starches, which are large polysaccharides present in almost all non-animal foods, particularly in potatoes and grains. There is a lot of cellulose present in the food as well, but there is no enzymes in humans to digest cellulose. Next is the digestion of carbohydrates in the mouth. When food is chewed, it is mixed with saliva, which contains the digestive enzyme ptyalin, which is an alpha amylase, secreted by the parotid glands. These enzymes hydrolyze starch into disaccharide maltose and other small polymers of glucose that contain 3 to 9 glucose molecules. Next, we look into the digestion of carbohydrates in the small intestine. First phase of digestion occurs by pancreatic amylase. Pancreatic secretion, like saliva, contains a large quantity of alpha amylase that is almost identical in its function with alpha amylase of saliva, but is several times as powerful. Therefore, within 15 to 30 minutes after chyme enters from the stomach into the duodenum and mixes with pancreatic juice, virtually all carbohydrates will have become digested. Next is the hydrolysis of disaccharides and small glucose polymers by intestinal epithelial enzymes. The enterocytes lining the villi of the small intestine contain 4 enzymes, which are lactase, sucrase, maltase, and alpha dextrinase, which are capable of splitting the disaccharides lactose, sucrose, maltose, plus other glucose polymers into their constituent monosaccharides. Here we see a schematic of how exactly the digestion of carbohydrates occur. The first starches are digested using ptyalin, which is present in the saliva, which digests around 20 to 40%, and pancreatic amylase, which further hydrolyzes 50 to 80% of the starch, which is then broken down into maltose and 3 to 9 glucose polymers. Next, maltase and alpha dextrinase present in the intestine act on the maltose and the other glucose polymers, dividing it into glucose. Similarly, lactose is broken down by lactase in the intestine into galactose and glucose, and finally sucrose is broken down by sucrase in the enzyme into fructose. All of these can be further converted back to glucose for efficient absorption and usage by the body. Next is the digestion of proteins. Proteins of the diet, the dietary proteins are chemically long chains of amino acids bound together by peptide linkages. The characteristics of each protein are determined by the type of amino acid present in the protein molecule and by the sequential arrangement of these amino acids. First is the thing we look into the digestion of proteins in the stomach. Pepsin, the most important peptic enzyme of the stomach, is most active at a pH of 2 to 3 and is inactive at a pH above 5. Gastric glands secretes large quantities of hydrochloric acid, This hydrochloric acid is secreted by the parietal or the oxyntic cells in the glands, which makes pH of stomach around 2 to 3. Next is the digestion of proteins by the pancreatic secretions. Most protein digestion occurs in the upper small intestine, in the duodenum and jejunum, under the influence of proteolytic enzymes from the pancreatic secretions, trypsin, chymotrypsin, carboxypolypeptidase, and proelastase. Both trypsin and chymotrypsin split protein molecules into small polypeptides. Carboxypolypeptidase then cleaves individual amino acids from carboxyl ends of the polypeptides. Proelastase, in turn, is converted into elastase, which then digests elastin fibers that partially hold meat together. Next is the digestion of peptides by peptidases in the enterocytes that line the small intestinal villi. The enterocytes that line the villi of the small intestine have a brush border that consists of hundreds of microvilli projecting from the surface of each cell. In the membrane of these microvilli are multiple peptidases. Two types of peptidase enzymes are especially important, which are aminopolypeptidase and several dipeptidases. They split the larger polypeptides into tripeptides and dipeptides and a few amino acids. These are easily transported through the microvillar membrane to the interior of the enterocytes. Again, we have a flow chart of how proteins are digested by the body. First, proteins are acted upon by pepsin, which are followed by which breaks it down to proteases, peptones, and polypeptides. Next, these are being acted upon by trypsin, chymotrypsin, carboxypolypeptidase, and proelastase, which break them down to polypeptides and amino acids. And finally, peptidases break all of the polypeptides down to constitute amino acids. Next, we look into the digestion of fats. The fats of the diet are by far the most abundant fats of the diet are neutral fats, which are triglycerides consisting of glycerol and 3 fatty acid chains. Neutral fat is a major constituent in the food of animal origin, but much less so in plant origin. In usual diet, there are also phospholipids, cholesterol, and cholesterol esters. First, we look into the digestion of fats in the intestine. A small amount of triglyceride is digested in the stomach by lingual lipase that is secreted by lingual glands in the mouth and swallowed with saliva. This amount of digestion is less than 10% and is generally not very important. Next is the emulsification of fat by bile acid and lecithin. The first step in the fat digestion is physically to break the fat globules into very small sizes so that the water-soluble digestive enzyme can act on the globule surfaces. This process is called the emulsification of fat, and it begins by agitation of stomach to mix the fat with products of the stomach digestion. Then most of the emulsification occurs in the duodenum under influence of bile, the secretion of the liver. Bile does contain a large quantity of bile salts as well as the phospholipids lecithin. Both of these, but especially lecithin, are extremely important for emulsification of fat. Next, we look into the digestion of triglycerides as pancreatic lipase. By far, the most important enzyme for digestion of triglycerides is pancreatic lipase, present in enormous quantities in a pancreatic juice, enough to digest within 1 minute all triglycerides it can reach. In addition, the enterocytes of the small intestine contain still more lipase, known as enteric lipase, but is usually not needed. Next, we have the end products of fat digestion. Most triglycerides of the diet are split by pancreatic lipase into free fatty acids and 2 monoglycerides. We have also a flowchart for digestion of fats as well. Fat, after action of bile and agitation, is transferred into emulsified fat on which water-soluble enzymes can work on. And on this emulsified fat, pancreatic lipase acts in order to form the fatty acids and 2 monoglycerides. Next, we have the basic principles of gastrointestinal absorption. First we look at the anatomical basis of absorption. The total quantity of fluid that must be absorbed each day by the intestine is equal to the ingested fluid, which is around 1.5 liters, plus that is secreted in the various gastrointestinal secretions, which is about 7 liters. This comes to a total of 8 to 9 liters daily. All but 1.5 liters of this is absorbed in the small intestine, leaving only 1.5 liters pass through the ileocecal valve into the colon every day. The absorptive surface of the small intestine is the mucosal villi. The figure demonstrates the absorptive surfaces of the small intestine mucosa, showing many folds called valvulae conniventis or folds of Kekri, which increase the surface area of the absorptive mucosa about 3 folds. These are the folds we are talking about. Also located on the epithelial surface of the small intestine, all the way down to the ileocecal valve, are literally millions of small villi. This project about 1 millimeter from the surface of the mucosa. The presence of villi on the mucosal surface enhances the total absorptive area another 10. Finally, each intestinal epithelial cell on each villus is characterized by a brush body consisting of as many as 1,000 microvilli protruding into the intestinal channel. This increases the surface area exposed to the intestinal materials at least another 20-fold. Thus, the combination of folds of Kekri, the villi and the microvilli increases the total absorptive area of the mucosa perhaps 1,000-fold, making a tremendous total area of around 250 or more square meters. Here we have a functional organization of the villus. This is a longitudinal view that is shown, in which we can see the central lacteal, the blood capillaries, these are the arterial ones and these are the venous ones, the vein and the artery that supply to the villi itself. When cut cross-sectionally, we will see a particular representation such as this. And here we can again see the brush border, the basement epithelial membrane of the villus, the arteriole, the venules, and the capillaries that are present inside the basement membrane, and also the central lacteal. Next, we look into the absorption in the small intestine. Absorption from the small intestine each day consists of several hundred grams of carbohydrates, 100 and more grams of fat, 50 to 100 grams of amino acids, 50 to 100 grams of ions, and 7 to 8 liters of water. Absorptive capacity of the normal small intestine is far greater than this, as much as several kilograms of carbohydrates per day, 500 grams of fat per day, 5 to 700 grams of proteins per day, and 20 or more liters of water per day. So we look first into the absorption of water. Water is transported through the intestinal membrane entirely by diffusion. Furthermore, this diffusion obeys the usual laws of osmosis. Therefore, when the chyme is dilute enough, water is absorbed through the intestinal mucosa into the blood of the villi almost entirely by osmosis. Next, we have the absorption of ions such as sodium. 20 to 30 grams of sodium are secreted in the intestinal secretion each day. In addition, the average person eats 5 to 8 grams of sodium each day. Therefore, to prevent a net loss of sodium in the feces, the intestines must absorb 25 to 35 grams of sodium each day, which is equal to about 1/7 of all sodium present in the body. The motive power for sodium absorption is provided by active transport of sodium inside the epithelial cells through the basal side walls of these cells into the paracellular spaces. This is demonstrated by heavy red arrows that are shown right here. The active transport obeys the usual laws of active transport, that is, it requires energy, and the energy of the process is catalyzed by appropriate adenosine triphosphate enzymes in the cell membrane. Next, we have the absorption of other nutrients. First, we look into the absorption of carbohydrates. Essentially, all carbohydrates in the food are absorbed in the form of monosaccharides. Only a small fraction are absorbed as disaccharides and almost none as larger carbohydrate compounds. By far, the most abundant of absorbed monosaccharides is glucose, usually accounting for more than 80% of the carbohydrate calorie absorbed. Glucose is obviously transported via the co-transport mechanism that we have discussed before along with sodium. There are two stages in the transport of sodium through the intestinal membrane. First is the active transport of sodium through the basolateral membrane of the intestinal epithelial cells right into the blood, thereby depleting sodium inside the epithelial cells. Second, decrease of sodium inside the cells causes sodium from the intestinal lumen to move through the brush borders of the epithelial cells to the cell interiors by a process of facilitated diffusion. That is, a sodium ion combines with the transport protein, but the transport protein will not transport sodium to the interior of the cell until the protein itself also combines with some other appropriate substance such as glucose. Fortunately, intestinal glucose also combines simultaneously with the same transport protein, and then both sodium and glucose molecules are transported together to the interior of the cell. Next, we'll look into the absorption of proteins. As explained earlier, most proteins after digestion are absorbed through luminal membranes of the intestinal epithelial cells in the form of dipeptides, tripeptides, and a few amino acids. The energy for this transport occurs by transport is supplied by a sodium co-transport mechanism in the same way that sodium co-transport of glucose occurs. Finally, we have the absorption of fats. When fats are digested to form monoglycerides and free fatty acids, both these digestive end products first become dissolved in the central lipid portion of the bile micelles. Because of the molecular dimensions of these micelles are only 3 to 6 nanometers in diameter, and because they're highly charged exterior, they are soluble in chyme. In this form, the monoglycerides and free fatty acids are carried to the surface of the microvilli of the intestinal cell brush border and then penetrate into the recesses among the moving, agitating microvilli. Here, both the monoglycerides and the fatty acids diffuse immediately out of the micelles and into the interior of the epithelial cells, which is possible because the lipids are also soluble in the epithelial cell membrane. that was all we had for today. These are the references for these particular slides, which are the chapter 62, 63, and 65 from the textbook of Medical Physiology by Arthur C. Guyton, the 11th edition. I thank you for listening to this video.